Good morning, dear students. Today we are going to learn about gene expression. What is gene expression? Means gene is expressing what is present inside a gene. Some genetic information will be there. And as it is expressing what is forming some products especially polypeptide chains. We know that polypeptide chain itself later will form some proteins, and that proteins can be some enzymes. And without any enzymes, it is not possible to carry out any of the metabolic pathways in our body. Without any metabolic pathway, there is no life. So, gene expression is very, very, very important. So, in this context, we need to understand, we need to learn the contribution of an eminent scientist with regard to gene expression, that is Archibald Gerald. What is his opinion? He proposes that whenever some metabolic pathway become abnormal, cellular metabolic pathway become abnormal, that may result some hereditary disease. And as this is because of some abnormal cellular metabolism, the main reason will be a defective enzyme. So defective enzyme cause that defective metabolism, and that ultimately result in a hereditary disease. In this regard, he studied one metabolic process, that is the metabolism of phenylalanine and tyrosine. He extensively studied the case of this metabolic pathway, and he postulates, he comes to this conclusion. So once inside the body, this amino acid phenylalanine, it can go through three pathways or three fate are there for this one phenylalanine amino acid inside a cell. Either it can be utilized for some protein synthesis as it is an amino acid, or it can be converted to another amino acid tyrosine, or else it can be converted to phenylpyruvic acid. These are the three pathways that it can go through. So, but by somehow, if any error or defect happened to some enzymes that is very necessary for any of these pathways, that may result in some errors. Errors means some hereditary disease conditions. And what all can be the disease in the case of this phenylalanine and tyrosine? The common diseases might have happened are alkaptonuria, phenylketonuria, and albinism. So we are going to discuss and understand about each disease. What is the condition? What is the error happened there? First, we can consider the case of alkaptonuria. In the case of this disease, two amino acid like phenylalanine and tyrosine tyrosine in its metabolic pathway, another component is forming as an intermediate component. Its name is homogenetisic acid. Homogenetisic acid. And this homogenetisic acid need to convert again to another compound, that is malayl acetic acid. So this is the reaction need to happen inside the body. And for this conversion, required an enzyme or the homogenetisic acid oxidase. Homogenetisic acid. In short form, I am writing it as HAO. Homogenetisic acid oxidase. This enzyme is defective in that situation. In which situation? Alkaptonuria. Alkaptonuric person, this enzyme will be defective, and this results in the accumulation of this acid in his or her body, and it will eliminate to the urine, and the urine will turn to black color. What color? Black color. So the main culprit here is the defect or a defective enzyme, homogenetic acid oxidase. So that is that condition. Then what about phenylketonuria? Phenylketonuria is also relating with the same amino acid, phenylalanine and tyrosine itself. Here the phenylalanine, in this case, the problem mainly occurs when phenylalanine, we know, is an amino acid that we are taking through our food, and there's also one enzyme. Its name is phenylalanine hydroxylase. So what's his name? Phenylalanine hydroxylase is the enzyme required to convert this phenylalanine to tyrosine in its metabolism, or in that metabolic pathway this enzyme is required. But in this type of disease condition or persons with this hereditary disease, this enzyme will be defective, and this conversion will not take place. And this phenylalanine and also another derivative, phenylpyruvic acid, phenylalanine and phenylpyruvic acid, they both become accumulated in brain, specifically in the cerebrospinal fluid, in blood, and also a portion will eliminate through the urine. So that is the condition in the case of phenylketonuria. So then next we need to understand about albinism. What is happening in the case of this disease? Here we need to understand that the problem emerge as the body try to metabolize tyrosine amino acid. Here the enzyme required is tyrosinase enzyme to convert the tyrosine to dihydroxyphenylalanine. So for converting tyrosine to dihydroxyphenylalanine, here required an enzyme tyrosinase, and this dihydroxyphenylalanine will later get converted to melanin. And we know that melanin is responsible to give color to our skin, hair, everything. So as this enzyme is lacking, then this further reaction will not happen, melanin will not produce, and those persons will develop a light skin tone. So that is the albinism. So these are the three diseases. All these three diseases emerge out because of some defect in enzyme that are necessary to carry out some important metabolic pathway. So defect in that or a defective metabolism, cellular metabolism, in that way result in some hereditary disease. So that is the thought. That is the idea or contribution of Archibald Gerald. Is it clear? Next, we are going to understand about one gene, one enzyme hypothesis. This hypothesis is very, very, very simple. Hypothesis means it is only a postulation. It can be right or wrong. So, what is that hypothesis? This hypothesis states that one enzyme, it is produced by one gene. From one gene, one enzyme is forming. So that is this hypothesis. One gene, one enzyme hypothesis. And who proposed this idea? This idea is proposed by George Beadle and Edward Tatum in 1941. And the hypothesis, as we illustrate, is like in this way. From a precursor substrate need to form an end product named C, and it is forming through a series of steps, and each step result in the formation of two products A and B. So that's what this precursor first changed to A, then it is becoming B, and then it becoming the final required product C. And these three steps, each step is catalyzed by separate enzyme, and each enzyme is produced or encoded by separate enzyme. This step is catalyzed by this enzyme, it is encoded by this gene, and this step is catalyzed by this enzyme encoded by this gene. So that is what this one gene, one enzyme hypothesis. From one gene or this a single gene is encoding a single enzyme. And even though this George Beadle and Edward Tatum proposed this idea, it is anticipated before them by another scientist. So he is considered as the founder of this one gene, one enzyme hypothesis. Then who was that founder? His name is Franz Moas. He anticipated this hypothesis. From one gene, one enzyme is forming. So I hope it's clear. But actually saying it's not the right thing basically. Actually, the right one is one gene, one polypeptide hypothesis, and this is the right one so far in the science. So then, what it means here, according to this hypothesis, what it means? Most of the enzyme consists of more than one polypeptide, and each polypeptide it is encoded by a single gene. A single G. A single G. So, this is what this hypothesis is explaining, and this is correct according to our modern science. Modern science is accepting this one this one compared to the other one. Other one means one gene, one enzyme hypothesis. Okay, so example that was taken to understand this hypothesis are one, tryptophan synthase is an enzyme found in Escherichia coli. It contains four polypeptides, and each polypeptides, not each, two polypeptides each is synthesized by two separate genes, like 2 alpha polypeptides encoded by which gene? TRPA gene. Likewise, 2 beta. So, 2 beta polypeptides encoded by TRPB gene. So, that is what in this enzyme for polypeptide. 2 alpha and 2 beta are present. That alpha polypeptides are encoded by TRPA gene. Beta polypeptides are encoded by TRPB gene. And likewise, here quoting another example also, that is our human structural protein, hemoglobin. Hemoglobin also contains 4 polypeptides, alpha, beta, likewise. That alpha set is encoded by one gene, and beta polypeptides are encoded by another gene. So that is what according to this hypothesis, and this is the more accurate so far in science. Okay. Next, we are going to learn about the central dogma. It's very, very important. It has asked so many times for your university examination. So don't omit this in this answer. Central dogma of molecular biology. Central is the main fundamental thing in the molecular biology. What is that fundamental thing? From DNA, based on the information stored in the DNA, a protein or enzyme is forming. But DNA directly cannot form the protein. From the DNA, A protein cannot form directly. It needs a mediator to work in between this DNA and this final protein. What is that intermediate? That intermediate or that mediator is an RNA, that is mRNA. So, from DNA, a protein is forming through the intervention of a mediator molecule, mediator nucleic acid, that is mRNA. So, that is what telling according to central dogma of molecular biology. It is explaining the interrelationship between DNA, RNA, and protein. So that is central dogma. It is or it explains the interrelationship between DNA, RNA, and protein. And its picture is shown here. From the DNA, RNA is forming through the process transcription. So, from DNA, how RNA is forming? Through transcription. And from RNA, based on the sequence, nucleotide sequence in the RNA, through translation, a corresponding protein is forming. So this is the series, this series of interrelation between DNA, RNA, and protein. That is central dogma. And here, look here. Here, a reverse pathway also possible to happen. That is done by a different enzyme. That also I am going to tell you next in the next slide. Anyway, I hope that central dogma is clear. In the relationship between which all things, DNA, RNA, and protein. Okay. Then we are going to understand about central dogma reverse. Now we have seen that from DNA, RNA is forming, from RNA, protein is forming. That is the direct, is the normal pathway. But here we are going to see that a reverse pathway also possible with the help of an enzyme, reverse transcriptase. And this enzyme is extensively understood and studied in RNA tumor viruses. And from an RNA, this enzyme can synthesize DNA. Clear? From an RNA, this enzyme, which enzyme? Reverse transcriptase, can synthesize a DNA. So this is the reverse of central dogma. In the central dogma, we have seen that from DNA, RNA is forming. Here, with the help of this enzyme, what is forming? A DNA is forming. So this enzyme is otherwise called RNA-dependent DNA polymerase. It is called RNA-dependent DNA polymerase. So that is central dogma reverse. And who isolated this essay for the first time? Howard Temin and David Baltimore. Look that, take care that name, Howard Temin and David Baltimore, as they found that after their name, this process central dogma reverse is otherwise called Teminism. So for your examination, some chance such a 1 mark question came, don't get confused. It is central dogma reverse itself. You need to write about the reverse transcriptase enzyme and what it is doing. What it is doing? It is forming a DNA from RNA. So that enzyme is called the RNA-dependent DNA polymerase. Next, we are going to understand about collinearity of genes and gene products. Collinearity. What is it? Collinearity between genes and gene products. We know that gene products are proteins. And what's that relation? What it is explaining here? Look, this is DNA. And in this DNA, we know that is composed of nucleotides. This is one, this is another nucleotide, this is another one in this way. And based on the nucleotide sequence in the DNA, in the protein, corresponding amino acids are added up. So that is collinearity. Clear? The sequence of amino acid in a protein, it has some relationship with the sequence of nucleotide in the DNA from where it get created, it get formed. So that is collinearity. Suppose if we consider this enzyme, this amino acid in this polypeptide chain, it is added here mainly because of which codon is present here. Codon means what? Three nucleotide form one codon. So based on that codon, each amino acid get added up in the polypeptide chain. So that means that there is a relationship, a collinearity, a correspondence between DNA and the amino acid sequence in the polypeptide chain. So that is collinearity. Each amino acid in the polypeptide chain is specified by a corresponding triplet codon in the DNA. So that is collinearity. I hope that the whole thing that we discussed and learned in the today's section is clear to you. So I'm going to wind up today's section. Thank you.
I'm an icon, man. I'm a girl about a 10. I should fuck her in a Benz.